All right, we'll start again. So welcome, everybody. My name is Beth Foss, and I am the operations director here at the Choroideremia Research Foundation. We're awfully glad that you're here with us on a very hot and sultry, humid day in the mid-Atlantic, sitting in the air conditioning. Hope you are too, if it's hot where you are. We want to make sure that we thank our sponsor, 4DMT, for helping us host all of these great webinars with researchers, such as Dr. Chen, who's here with us today. I have to tell you, when Dr. Chen, you and I had spoken probably 2 weeks ago in preparation for this webinar, it was new science to me, and it was really exciting, exhilarating, compelling. But I always say, okay, we just need to keep things in perspective. And so it was extremely exciting. But I just wanted to welcome you here. And as always, everyone, I will be taking the questions and answers and the chats. I'll be monitoring the question. And I'll ask at the appropriate time if I can break into Dr. Chen's presentation with a slide, or we'll wait to the end. We'll get your questions answered no matter what. So Dr. Chen, tell us a little bit about yourself, and then I'll just turn off my camera here.
Thank you.
Sure.
So I'm a associate professor at the Harvard Ophthalmology, and my research involves optic nerve, really focusing on optic nerve diseases, degeneration and neuroregeneration. I've been working on this new approach of bioelectricity, especially using a non-invasive approach for ocular therapies for in the past maybe 5 to 7 years.
Wonderful. Wonderful. Well, thank you again for carving the time out of your week to spend it with us. And we go ahead and get started.
Thank you. First, I want to thank the organizer to inviting me and giving me the opportunity to share this research. So I hope that everybody can see it right now, and I'm putting it on the
great
presentation mode. Okay. Yeah. Cool. Yeah. So this is a new. Well, I would say new. It's relatively new to the effort in my past research career. But we've been working on this with 5 to 7 years in the past. So this is my disclosure, just for the sharing the purpose. And I believe that many of you coming here for this talk because of your interest of choroideremia, which is rare genetic linked, X-linked retinal degeneration diseases. But it actually belongs to a larger category of retinal disease, or we usually call it inherited retinal degenerations. And this type of disease actually are major cause of visual loss in young adults and children. The number actually says that there's maybe every 1,000 people are affected by this genetic eye diseases. And maybe 50%, 30 to 50% of these patients are affected only by one single gene mutation. So not only like choroideremia belongs to such a group of disease, actually this group of disease patients also share many common features of vision loss or the cause of vision loss. So many of these patients will start by night blindness and then gradually loss of vision, and usually from peripheral to central. And these are normally the results of deterioration or death, loss of photoreceptor cells, and sometimes could also be retinal pigment epithelium cells. And even within the family members, and we notice large variations between person to person in terms of rate and degree of vision loss. And therefore, there's always a theory behind and say it's not only about the gene mutations that cause the vision loss or photoreceptor cell death, but it's also the environmental factors and as well as inflammation that may be involved in this process. The reason I'm talking about this is because these are really impact on how we think about the disease, and currently there's really no treatment except maybe gene therapy has been approved of only one very special type of inherited retinal degenerative diseases. But for generally, the doctor will just prescribe or actually recommend wearing sunglasses and taking dietary supplements such as vitamins and omega-3. And gene therapy and stem cell therapy are I would say, outlook for the future that we're actively working and trying to develop. But on the other hand, we're thinking that takes time to develop this therapies to really stop or reverse the vision loss. And so one of the things come to our mind is, can we find other ways and even less invasive to prevent or slow down the vision loss? And if it's possible, to reverse it a little bit even. So that comes to our mind is when we think about these diseases, we're primarily thinking about these are neurons that are dying, neurons that sense the lights that are dying. So neurons normally communicate with each other as well as with the rest of our body through electric signals. So we can say that electricity or electric signals are the language neurons speak to each other, as well as neurons speak to the rest part of our body. And therefore, the question would always be floating in our mind is, can we talk back to neurons through electric signals? Or can we read the mind of these neurons through the electric signals? And the fact is, we are already being able to at least using some tools to read these electric signals, such as when the patients go to see ophthalmologists and eye doctors, and they will use electroretinogram to read the functions of the retina. And same thing, like we're using the EEG to read the functions of the brain, or try to detect the disease of the brain. But more than that, there are actually more efforts of trying to develop, let's say, prosthetics or electronic devices to both invasively or non-invasively to treat the neurodegenerative disease, neuropsychiatric diseases or neuropathic pain, as well as we'll know like neuroprosthetic try to replace limb, the loss of functions. And there are also effort in trying to use electricity to treat or at least manipulate ocular or eye diseases. And that's because eyes basically functions like a mini brain and structures like a mini brain. So when we receive the visual signals coming into the eye, the retinal cells will convert that into electric signals and then pass on to the brain. And that's how we see. And that's how retinas talk to the brain and talk to each other. So basically, this is a rough structures just to show that how other of tissues, if you can see it, that's actually the retina. I hope you can hear me okay. It says my internet connection is not stable. So although this is a very thin layer of tissues, it's actually contain a very complicated neuronal structures and at least 7 different types of major neurons, and they can even be subdivided into smaller subpopulations. So, and as I mentioned, that these cells communicate through between each other and also to the brain through electric activities. And the idea is, can we try to find out what is the optimal electricity or bioelectricity that we can talk back to those neurons and probably help supporting the health and functions of those neurons. And so this is really when we begin to think about this I would say several years ago, and these are the scientists that at the time in my lab that and begin to think about the same line of the therapeutics or approaches. So especially Eric Yu is the first really research fellow in my lab and decided to put his hands on the on testing how electric activities may be able to improve the visual function. And Tor is our collaborator from Oslo University, and through collaborating with him, we actually obtained quite a large fund from Oslo University and Research Council of Norway, and that's really supporting us and allow us to perform all the studies I'm going to talk about today. And Karen and Kinson Chow are really the key drivers through all these years. And so we're setting up both the animal models to testing this in mouse primarily, and as well as also putting the cells in culture and directly giving the electrical stimulation to the cells and to further understand how these individual cells respond to the electricity. So, and quite exciting is that when class started to work on, we decided to use this inherited retinal degenerative disease mouse model. And at the beginning, we used rhodopsin knockout mice, which means mice lacking a gene called the rhodopsin. And that's actually light sensing molecules in the photoreceptor cells. When this gene is missing, this actually, the mutation of this gene is also the most prevalent mutations that cause inherited retinal degeneration. That's one of the reasons we decided to start from this mouse model. And at the beginnings, we were not sure what is going to happen. Although there have been like clinical trials that done here and there in the different part of the world. Really very little study has been done in like science or in the lab, and to see how exactly the electricity works. And especially we do not know whether a mouse can respond to the electricity. But quite exciting that when Dr. Yu started to acquire a machine from a company who have done this in human and shown that at least it's safe to really perform the electric stimulations. And when we give it to mice only a week after, then we're already able to see the improvement of the visual response from this mice. And what I'm showing here on the lower corner, if you'll be able to see, left corner is this green staining actually stains the photoreceptor cells. And so you can see that the left side is the control, which means we did not give the electricity to the mice. And then you can see there's this green functional cone cells already been largely degenerated. You can only see one line of residue cells survived there. But then on the right-hand side is ones that mice receive the electric stimulation. You can see a robust increase of the thickness of the screen stained layers. So we did quantifications of counting the cells and measure the gene expression levels of those cells, and you can all see that this orange bar is showing that there was significant improvement of both the survival and the number of these cells. So at the time, I'm thinking why not. Okay, yeah, so at the time that we acquired the machines, and that's what probably most people use in clinic to stimulate the neurons, because we call that rectangular waveform. So this basically looks like straight lines, as I'm showing on the top left here. So we were wondering that do neurons respond to different shape of electric stimulations? So if we gave maybe a sine shape or the ramp shape, as I'm showing on the first line, would neurons respond differently? So this is another fellow in the lab and decided to take eyeballs and just putting the this oscilloscope here and try to record the shape of electricity. So we give it to the eyeball, and then this is what he recorded from. And basically saying that when we gave this electric, this kind of shape of electric stimulation, and normally the retinal tissues will receive the very similar or response were very similar electric, when we trying to look at how efficient this electricity can penetrate eye tissues, we found that actually one waveform, which is this triangle kind of waves, penetrate the tissues very efficiently. And at a very low voltage, we can already detect the current at the back of the eye. So suggesting that this may be a more efficient way of develop or deliver electric field to neurons. And therefore, another fellow joined my lab and decided to test, compare different electric waveforms in mice. And so just to see which one actually gave us a better improvement of vision in this mice carrying inherited retinal degeneration. So this is a schedule showing that he gave this electric stimulation every day, and then for 7 days, and he compared the rectangular waveform and the ramp waveform. And so we use the visual acuity test, which basically asks the mouse to read, but they only read the black and white lines. And then we also record electric activities, as I mentioned, that we actually can use that to read the health of the neurons. And then you can see that in all these categories, this black bar, which means mice receive the ramps electric waveforms, have the best performance. They really shows a very good preservation of vision, both electronically or when we ask them to read, we also read the best.
Dr.
Then,
Chen,
please.
just real quickly,
Yeah.
if you could please maybe turn your volume up just a tad.
Okay.
And there is some just choppiness in the, and I know that you said that you were having some problems with your internet. Sometimes that happens with some of our other presenters and they stop using their camera. You know, if you turn your camera off, maybe that will give you a little bit more oomph.
That's a good idea. Okay, I hope it's better now.
That is better, yes.
Okay, great. Yeah.
Thank you.
Sure, you're very welcome. And also, please feel free to ask me questions. And otherwise, I'll just continue.
Yeah, perfect.
Thanks. So, so then when we found out, we have done a whole series of experiment. I mean, I'm just skipping some of the in vitro or culture studies, but in this studies, we actually found the ramp waveforms, the triangle waveform, give us the best results in terms of improving vision. And then my fellow decided to look at and say how frequent we have to give the electric stimulations. Let me see. Yeah, and therefore he designed this electric stimulations like by 3 groups. One is every day, the second group is every other day, and the third group is every 3 days. And then he found out actually every other day or every other day both gave up really good results of preserving vision. But if we skip 2 days and every 3 days, and the results was lost, which means it's probably best to have the electric stimulation maintained every day. And this is only 4 minutes per day in a mouse test, which is also quite convenient as we thought. And if even someone skip a day, it probably doesn't matter. But and apparently persistent stimulations every day will give us the best results. And we're really building this and trying to understand if we move on this to humans, what should be the frequency and what should be the time durations of treatment. And so the previous studies, we're looking at electric retinograms, means the electric activity of the neurons. And here we further check the quantitatively and looking at it, the magnitude of electroretinogram and also the the visual acuities and the visual contrast sensitivity. So the left-hand side basically is showing you the waveforms that we detected from the electric stimulated mice and non-stimulated. So you can see a far improvement of the electric response and health of neurons. And also the right-hand side really showing you how we ask the mouse to read. So when we change the width of this black and white bars, if mouse can still read and they will respond to it. And in this way, you can see that the mice treated with electric stimulation like shows much better abilities, or they can read much finer lines in this response. And in addition, not only we found that, previously, the previous slides have shown that electric stimulation keep the neurons survive better. And actually we found not only they survive better, they're also putting out more process. So if you may be able to see there's more lines in here and compared to the ones on the left-hand side, which are without electric stimulation. So this more sprouting or nerve growth would also means that the neurons will connect more broadly to internally or talk more frequently to other cells inside the retina, and therefore giving us more sensitive light response or vision. And here is just to showing you different layer of neurons and where they're correlating in what you're showing here. So as I'm talking up to now is we're all talking about the behavior of neurons because we done neurons respond to electricity. However, there are also other type of cells in the retina. They're supporting cells, and we normally call them glial cells or non-neuronal cells. And they are the cells that maybe standing out, actually having process extending out the entire retina, or maybe just floating around and checking the health of neurons. So it's important to also keep those cells happy and healthy. And therefore, we're very curious that how these cells will respond to the electric activities. And we normally can now read their behavior through the ERG. And therefore, we decided to take them out in the culture dish and just to see how they respond to the electricity. The original idea is, since these are non-neuronal cells, they're not supposed to, or at least not robustly, respond to electricity. But to our surprise, when we isolate those cells and put them in culture, and that will, in the middle, actually shows how they look like in the culture dish. And then we gave them electric stimulations, and after that, we're looking at what gene has been changed to respond to electric activities. And we found that the electricity does change the gene expressions of the cells. And they're not only change the gene expression in neural cells, but they also change the gene expression in glial cells. So those both are non-neural cell types. And looks like what happens is this gene expression changes, causing the cells to become more calm and less inflamed. So, so in another word, to make them healthy, happier or healthier. And this is actually the 3D slides images that my fellow Dr. Lenikov obtained to show how the microglia actually looks like inside the retinas. And I just thought they're so beautiful, so I'm sharing that with you here. So in addition, Dr. A.J. Ashok also found that not only the electric activities, bioelectric activities, change the gene expression, they could also change or acting on the we call epigenetic level, which means without changing the gene itself, but by modifying the modifiers on the gene, and to control their expressions. So electric stimulation is really acting on multiple levels to regulating neural activity as well as glial activities. And by far, it really looks like they're driving the cells toward a more healthy and homeostatic status. So, so one of the questions before we move this to human is we wanted to know that where should we stimulate the eye? I mean, when we put in the culture dish or when we first try it, we just put it on non-invasive because we want to uh, make it easier to be applicable to human. And however, there are many different ways some people or scientists trying to stimulate the eyes. So what I'm showing here, like each arrows point to a position that actually have been tried by scientists to stimulate the eye. And the most common one that are used in clinic, but it's also the minimally invasive, are the transorbital, which is stimulate the top of the eyelid, on transcornea, which means directly put the probe on the cornea, on the eye, right? And then transpalpebral, which means only on the eyelid and very precisely. So we decided that to compare and see which method give us the most least harmful but most beneficial effect. And so our collaborator, Dr. Meng Yang, who was working primarily in the anterior eye and also the cornea, and decided to test this in the mouse. So she put the electrode on the cornea, which we call transcornea, or on the eyelid, which it could also be transorbital or transpalpebral. And the green color means, so basically use fluorescence dye to stain the cornea and to see whether they got injured and how happy they are. And so green means they're happy and healthy, and red will means they probably are dry or not very happy. So as you can see that when we put electrode directly onto the corneas, it's actually causing some kind of dryness in the cornea. However, we put it on the eyelid, and this kind of sensations goes away. And this actually correlated with what many patients who received the transcornea stimulations in clinic and then found that they do develop dry eyes. So after this, I think we also proved that a transorbital and transcornea, this or a transpalpebral, which non-invasive electric stimulation actually give us the best results for rescue neurons and also least harmful for corneas and the surface of the eye. And so if I put together, just to summarize what we have found so far, and as well as by other people who work in the area, that we actually can conclude that the electrical stimulation can act on multi-factors in the retinas by improving the function of retinal neurons. This including improving the neuronal survival, stimulating the production of neurotrophic factors, which supply the retina. So,
Dr. Chen, could you repeat that one more time at the last statement, because it broke up quite a bit?
Yeah, it's actually just lead to this conclusion. So basically, that's what I'm telling everybody. And that one is a graphic form, and this is really the word form of what I just said. So I think we're quite happy to see that this non-invasive transorbital or transpalpebral electric stimulation can promote the photoreceptor survival and improve vision, as we show in the inherited retinal degenerative mice. And we also show that the effect of this electric stimulation are multifactorial. So it can enhance neuroplasticity, which means enhance neural connections with each other, and also promoting the growth as well, and probably via both genetic and epigenetic mechanisms. We also demonstrated, which is quite somewhat surprising, is bioelectricity actually present a beneficial effect to non-neuronal cells to suppress the neuroinflammation. And therefore, altogether, we would say that this electric stimulation may offer a safe and also very novel interventions for future therapy of the retinal degenerative diseases. And that we are very hopeful that, and also believe that this should be able to apply to broad terms of retinal degenerative diseases, including choroideremia, which we're very interested in collaborating and also specific tests in these mouse models of our paradigms. And at the end, I want to thank our the people in the lab who either the former fellows or current fellows who have contributed to this work. And you have met some of them through the pictures I've shown. And also our collaborator at the Schepens, Dr. Darlene Dart and Meng Yang. And especially thank you, Dr. Tor Yuthan from also university who continued and also supported our research. And these are some of the current members in the lab and continue working on the project. And thank you. So I think I'll stop sharing and then people can probably ask questions.
Wow, what a great group of kids, young people, I should say, young people in your lab. Thank you so much. Now, for everybody that was watching this who has choroideremia, this is pretty exciting, right? I mean, I know it's science, I know it's research, of course, but it's pretty exciting. So we do have some questions coming in. Are you ready? Here we go. The first one is, hi all. I hope let me just, it was a personal message to me. Okay. Is there an age limit, or should I say, if I only have a couple of degrees left of central vision, would I still have a chance of saving the little sight I have left with this process? He writes, I'm scared of losing the little bit of sight I have left. And like many of us, we desperately are trying to hold on to or keeping hope of what I have. Thanks for all of your work. This is somebody from Ireland.
Thank you. That's a very good question. And that is actually part of the work that is ongoing in the lab. And we're doing both and using I think today I'm focusing on using this non-invasive electrostimulation rescue, or which means to protect the vision loss, protect against vision loss. And other parts in the lab, we're also trying to use different approaches, including electrostimulation, trying to reverse vision loss, which means we're trying to regenerate those lost neurons and also regenerate the lost connections. And I'm happy to say that we have obtained some really exciting progress. And I think there is a hope that we could even reverse, not only prevent the vision loss.
Okay, here we go. I'm going to keep jumping through these because we have a lot of people here and a lot of questions. It says, thank you for your presentation. After how many days of no retinal activity or the beginning of the disease that you were having a result of improving vision? In other words, if the cells are dead or not used after a while, is it possible to make the cells function again?
Right, that's a very good question. That's also the most challenging part, as I mentioned, that what we're trying to do is once the vision is lost, which means that the cells are gone, can we reverse the vision loss? And so that really requires the regenerating of either the nerve connections or the neurons itself. And that is ongoing work in the lab. And I would say that we already have part of the success in animals. And so, and certainly this still will take years before this can be moved to human. And I would say that the hanging there, and I think we're getting there.
I told you that was going to be one of the questions I was asked as a timeline, but no one's actually, well, it is. There's a question and a couple down here, but we're going to go in order. Is this an AC or DC electricity?
It's AC.
Okay. Are interested patients able to access this type of therapy currently?
And I would say that there are, like different institutes are offering electric stimulation for ocular therapy, but I see the issue is it's not a standard electric stimulation paradigms, and that is the reason why we put in so much effort, try to standardize it and try to work out what is the best condition and most effective ways to deliver the electricities and to get maybe the best results. Yeah, so I would say that there are institutes available. The treatments are like being offered in a way like electric stimulation in general. The ways how people delivering is very safe, but it's less effective. So we're hoping that maybe moving what we have found so far using this defined paradigms to develop new treatments or more effective treatments.
The next question is the one that I was sure that was going to be asked. When will human trials start? What is the timeline that you think you're going to look at?
We are actively working with looking for collaborators and hoping to move this to clinical trials right now.
Okay.
That actually, there are already companies are working on the clinical trials that I'm collaborating with. It's just that I'm not specifically on this paradigms.
Okay. The next question. Says, and what voltage is used for humans?
As I mentioned that when we use different paradigms, then actually we can use a quite low voltage, which means to increase the safety or increase the safety range or profiles. At this point, I think we're using a micro. Well, we're really controlling the current, but the range will be at the millivolt microvolt range.
Okay. Next question. I think transorbital stimulation has been tried for AMD in the past. Do you know about those studies, and is there any similarities?
Yes, actually, there are active clinical trials on the even in the US for using the non-invasive electric stimulation for AMD right now. I'm hoping that there will be successful in the next couple of years, then it can be available for many people.
Wonderful. Another question. I have a 7-year-old who has been diagnosed and a 2-year-old who we are waiting to find out. How young is too young to begin therapy? Great question.
Yeah, well, at least when we tested in animals, I mean, it's actually younger is probably an advantage because then the cells are still I mean, you still have many cells functioning. And so yes, at this point, the paradigms we're using is really more toward rescuing them or save them from dying. And therefore, it's probably a good idea to start with the younger ages.
All right, we're gonna go to this. Excuse me one second. Okay, the next question. Here. Okay. Thank you very much. Thank you. What are the differences between your approach and that of a clinically available transcorneal electric stimulation devices? That's coming from a researcher.
Yes, I mean, that's why the scientists that's always trying to work out what is the best conditions and which one works the best. I think, as I mentioned, that there are clinical trials or actually treatment even being available or being offered by different institutions, especially outside of the US, for ocular diseases. But most of these treatments are not standardized. So the electric stimulation paradigms are not fixed, or as from what we see or from our tests in animals, it's not optimal. And therefore, we think there is need that with the run clinical trials to to standardize this electric stimulation paradigms and to ensure that we really get the most benefit out of the treatment.
It says, thank you, Dr. Chen. Could you please explain the heat map homeostasis slide again? It was at the time a little hard to understand. Thank you.
I think that it's talking about when we take the cells out, we put them in the culture dish, and then specifically just give the cells electric stimulation in the culture dish. And that's a method we're trying to understand how electricity acting on individual cells. And so in that studies, what we do is after we stimulate the cells, we take all their transcriptomes or RNAs and we sequence them. So we see which gene has been changed. So those heat map is trying to show that what gene has been changed compared to the normal conditions and also compared to the diseased conditions. Actually, I think the ones I've shown is already the published data. So we're showing that with the electrostimulation, the cells actually maintain a more homeostatic transcription profile, or they're more similar to the normal cells compared to the inflamed cells or diseased cells.
Nicely done. I think someone asked about, would this be safe to use on younger patients such as teenagers? And I think you answered that earlier. Here's another question. I am aware of a few startup companies offering electric stimulation for improving vision. Have you evaluated any of those products? Any thoughts or comments about proposed therapies?
Yeah, as I said, actually, the electrostimulation in general, I think mostly because it's already been used for treating muscle pains and even treating neuropsychiatric disorders. And therefore, these similar paradigms probably, and people are converting it and trying to treat ocular or eye diseases. I think the key is people are using very different paradigms and therefore get the very mixed results from the clinical testings. And some people may get improvement of vision and some people don't. For a long time, people don't know why. And then that's also caused suspicious and say, does the electric, especially non-invasive electric stimulation works? So through these studies in animals, at least we shown that electric stimulation could work if it's been applied in the right paradigms and right conditions. So I have not evaluated those product or treatment on the market. So I cannot say which one is good, which one is not. I can only say that the ones we tested and we compare different paradigms, and we know some should work, some may not.
Since all cells work using electric impulse, and the brain, for example, acts like a capacitor using lipids to slow down a current, could this process be creating a burst, but actually overusing what tissue is there, instead is causing wear, which speeds up deterioration in the long run? Ooh, I think they get an A+ for the question.
I hope I understand the question there. I think when that's part of the things why we take the cells out and put in the culture and trying to see how electricity directly acting on the cells and maybe what molecules or how cell membranes respond to it and what molecules respond to it. So at least with the paradigms we're using, and it's really looking like it's the ion channels that are buried on the cell membranes, especially voltage gated ion channels are the ones that actually most sensitive and first respond to the electricity and the potential. So I don't think our electricity or the ones that has been used now is can go that far to really burst the cell membranes, and that's why it will cause cell death. So, so there is a safety range of the electricities. How far can you go for current amplitude or voltage amplitude? Within that amplitude, I think it does not burst the membranes or cause deterioration so far.
Do you have time for more? Because we have quite a few more popping up.
Sure.
Okay, thank you. Thank you. Although it may be less effective than what you are trying to develop, would there be a risk or harm to using the existing products available in the clinics? Sort of answered that, I think.
Yeah, it's very hard to say whether for the long term. If it doesn't do you good, does that do you harm? Don't know.
Yeah. How will information about trials be provided to the public? Good question.
Yeah, I'm sure it will be published information, and we will, if we're doing it, or people are doing it, they want to invite the right patients and right people to participate in the trial. So it should be public available.
You know, I think that's one of the, I'll make a little plug right now, but we talked about that. Dr. Chen, the CRF, is going to be hosting our conference in Minneapolis next at the end of June. I believe it's June 27th. That will be on our website soon. But we're going to be having, I hope, someone from your lab that can come, as well as other research and scientists. And that really is one of the best places if you can join us in Minneapolis. And we will be putting things, of course, on our webpage, and clinicaltrials.gov is also, again, a good place to just write in choroideremia, and you can find out what trials are taking patients and so forth. But hopefully we can have you come in September, or June. All right, next question. How can I be a part of the trials? In Ireland, there's nothing happening, but I'm willing to travel. Thanks again for answering our questions. Exciting times ahead. Absolutely. I would agree. I think I answered that, unless you have more to
No.
to respond.
Yeah,
Okay.
perfect.
Okay, here's another one. Can you speculate on the mechanism of the increased cell function using this electrostimulator? Thank you. Or stimulation.
Yeah, that's an excellent question, and that's the part of the work being done in the lab and trying to decode how the electric stimulations work in neurons and non-neuronal cells. So as I said a little bit already, that we believe in this range of electric stimulations, it's probably acting through turning on the voltage gated ion channels that to regulating the ion signaling, which are very important signaling events happening inside the cells that tell the cells what to do and how to behave.
Hmm. And as you're in your lab and you're having these discussions with your team, how do you think that this stimulation would happen? Would it be a type of contact in a sense on the eyelids, or what kind of device are you thinking it would look like, possibly?
Yeah, there are actually already some FDA approved parts that so we're thinking that for the transorbital, basically can be even a sticker being stick onto the eyelid or on the forehead around the eye. And to develop which is electrically conductive, so they can convey the electric signals to the eye.
That sounds fairly easy. All right, here's another question. It says, to follow up on the already marketed products, how will we know when this and other credible therapies are openly available? Well, that would be.
Can you repeat that again? When?
Well, I think it's to follow up. How will we know when this and other credible therapies are openly available? And I think we sort of answered that already. Again, for all types of research and clinical trials, the best is clinicaltrials.gov, and you put in choroideremia. That can get you answers, and you can follow different trials currently happening. And again, just come to our website and explore, and you can put in research, and we'll be updating folks as it goes on. I hope that answers your question, Paul. All right, and then maybe out of the actual subject, but how do you get mice to show up with an IRD?
It's the same thing as that happens in human, because when mouse carrying a mutation in those genes that cause the human eye to develop IRD, mice will develop IRD too.
Are you looking at moving into other animals models?
We will, because if this one will go go to human trials. And the normal process is we can start with a mouse and rodent, which actually their eyes are very similar to their retinas is very similar to humans. However, because their size is small, and we still want to test in the larger animals before we move to the human trial. So for sure.
This is, I think, the final question, unless any more pops in. With this disease, oh, and somebody just did. With this disease, it seems the tissue of the retina does not receive maintenance, cell sloughing, or nourishment. How can stimulating those neurons be effective when the organ itself is turning ineffective? Pardon my lack of technical terminology. I think you're doing just fine with your questions. How would you respond to you want me to ask again?
If I understand correctly, then I think, yeah, the simple answer or late-term answer to it is electricity is the language neurons speak. So we're trying to speak them in a healthy language rather than a diseased language to keep them healthy.
Okay. I hope that helps. Here's the final one. Have you also explored, oh gosh, light therapy? It's a very long word that I'm not going to be able to pronounce. I apologize. Have you also explored light therapy as a possible therapeutic for IRDs? There has been some research to show that red and yellow wavelengths help prevent oxidative stress. Could this be used in combination with electric stimulation for a 1-2 punch?
We haven't tried it. I don't know.
All right. I think that's it. So let me see.
Great, thank you. Yeah, and great questions. I think that's the ways to keep the science going and keep us thinking hard and working hard.
Well, we're very, very grateful. Kathy, our executive director, too, just piped in and said, thank you so much for your time. And we're really grateful. Hopefully we can meet you in person next June. But until then, we just wanted to say thank you to your team. Please keep up the good work. And we just want to thank everybody who joined us today. Thank you for coming, and please keep your eyes on the emails because we have a good amount of additional webinars coming up with different researchers. So thank you all. Thank you, Dr. Chen. And thanks again to our sponsor, 4DMT. And everybody have a wonderful day. Bye-bye.
Thank you. Bye.
Bye-bye.